Good morning, everyone. Thank you for the invitation. My name is Dr. Saad, and I'm from the University of Sydney. Today, I will be talking about the effect of epigenetic regulation on chronic kidney disease. I will first define epigenetics and describe its role in the progression of chronic kidney disease, or CKD, and in fetal programming to CKD. We'll then present our data on the effect of epigenetic modulators on obesity and maternal obesity-related kidney disease, and talk about the clinical implication of our finding. So what is epigenetics? Epigenetics are heritable changes in gene activity that are not caused by changes in DNA sequence. Agouti mice are good examples of epigenetics. These mice are identical genetically, but as you can see, they look very different. So in normal situation, the agouti gene, which is responsible for the coat color, is methylated, and the mouse is brown and has low risk of disease. The yellow mouse, which has an epigenetic mutation, is fat and has high risk of disease. The effect of epigenetics are also clear in monozygotic twins. Even though identical twins share the same DNA, they aren't necessarily exactly alike. Environmental factors and epigenetic factors create differences as twins age. These factors explain why identical twins can be different, whether in appearance, temperament, or personality, and potentially why they can have different disease predisposition. The two main components of epigenetic changes are DNA methylation and histone modification. DNA methylation occurs by the addition of a methyl group to the DNA, which modifies the function of the genes and gene expression. DNA methylation is an integral process in normal human development. It typically occurs at CpG denucleotide sites via the action of DNA methyltransferase enzymes. When a CpG island in the promoter region of a gene is methylated, expression of the gene is usually repressed. Histone also undergo post-translational modification, including methylation, acetylation, phosphorylation, and ubiquination that affect chromosome structure, influence the accessibility of DNA to interacting proteins, and alters transcription. Epigenetic modifications are implicated in the development of chronic disease. DNA methylation is the most stable epigenetic mechanism and the best understood epigenetic modification in the context of kidney disease. It occurs in response to environmental stimuli, including diet, metabolic fluctuation, exercise, oxidative stress, inflammation, drugs, and toxins, and it can be modulated by the cellular milieu. As we described in our recent review, multiple studies have reported a link between DNA methylation, obesity, and diabetic kidney disease, supporting the role of DNA methylation in disease development and progression. However, to date, it's not clear whether DNA methylation can be used as biomarkers for chronic kidney disease progression, or whether drugs which modify DNA methylation can have a therapeutic role in chronic kidney disease. So can DNA methylation be used as a biomarker for diabetic kidney disease progression? As you may know, not all diabetic patients develop kidney disease, and not all who develop kidney disease progress to end-stage kidney disease or require dialysis. 30 to 40% of patients with diabetes develop kidney disease, but chronic kidney disease progression varies substantially from patient to patient, even among those with similar comorbidities. So can this be due to epigenetic differences? To determine whether DNA methylation can be a marker of diabetic kidney disease progression, we did a longitudinal study using a high-fat diet-fed animal model, which we have recently demonstrated to induce features of type 2 diabetes, including glucose intolerance and insulin resistance at 9 weeks, which represent adolescence, with significant renal structural damage and albuminuria by week 32, which represents adulthood. In this model, established chronic kidney disease is only demonstrated in around 40% of the animals at 32 weeks, as in human studies. Blood was collected from the same animals at 9 weeks and 32 weeks to assess DNA methylation over time, and kidneys were collected at 32 weeks to confirm the development of renal pathology. Animals fed chow diet were used as control. Assuming that 30 to 40% of the animals would develop progressive chronic kidney disease, animals were grouped into normal or non-progressive and progressive disease based on urinary albumin to creatinine ratio and urinary KIM1 values. So as you can see in panel A, the high-fat diet 2 group or progressive group had higher level of urinary albumin to creatinine and KIM1 compared to the non-progressive group or high-fat diet 1. Animals in the progressive group also had increased renal KIM1 protein expression and reduced levels of the antioxidant marker SOD2 compared to the non-progressive group. This was associated with increased renal DNA oxidation and significant changes in renal pathology, such as glomerulosclerosis, tubular and interstitial fibrosis, and tubular injury markers compared to the non-progressive group. The progressive group and non-progressive groups had different levels of 5-methylcytosine or 5-MC, a marker of global DNA methylation, and different levels of DNA methyltransferase DNMT1 and 3B, enzymes responsible for the maintenance and de novo DNA methylation. They also had different levels of 5-hydroxymethylcytosine or 5-HMC, an important intermediate in the active DNA demethylation process, and significantly less TET1, an enzyme responsible for DNA oxidation or demethylation compared to the non-progressive group. Using reduced representation bisulfite sequencing or RRBS, we demonstrated differential gene methylation in the blood of progressive and non-progressive animals at week 9, which means before the development of diabetic kidney disease, and at week 32. And then, as you can see here, hypomethylated genes are shown in blue, and hypermethylated genes are shown in red at the 2 time point. Common genes that are differentially hyper or hypomethylated in the 2 groups at the 2 time points are expressed in this heat map. So in brief, we identified 48 common genes, out of which 19 genes were consistently hypomethylated or hypermethylated over time. Pathway analysis for the regulated genes at week 9 revealed alteration in signaling pathways related to kidney development, structure, and fibrosis, suggesting relevance to the kidney and supporting the role of DNA methylation as potential biomarkers for diabetic kidney disease progression. Now that we demonstrated the role of epigenetic change or DNA methylation in disease progression, we wanted to determine whether epigenetic changes or disease predisposition is due to the in utero or early postnatal environment, and whether fetal programming can affect chronic kidney disease progression. Going back to the agouti mice, we know that when the pregnant yellow mice were fed a methyl-rich diet, most of the pups were born brown and stayed healthy for life, which shows that the environment in the womb influences adult health. We also know that nutrients from our food are funneled into a biochemical pathway that extracts methyl groups and then attaches them to our DNA. So diets that are high in methyl-donating nutrients, as example, can rapidly alter gene expression, especially during early development when the epigenome is first being established. This means that our health is determined by what we eat and by what our parents ate, and this can program the offspring to future disease. It is known that the birth phenotype can determine the adult phenotype and influence the risk of developing disease later in life. The prenatal environment can affect the birth phenotype through developmental disruption and epigenetic changes. The genotype or gene makeup of the individual can also affect the birth phenotype. In addition, the postnatal environment can also affect the adult phenotype to induce disease risk, and this can be amplified if there is a mismatch between the prenatal and postnatal environment according to the predictive adaptive response hypothesis. Critical period of development ranges from preconception to early childhood. Suboptimal concentration of an important factor during a critical period of development can lead to permanent structural changes in an organ. We have previously demonstrated that the offspring of obese mothers are insulin resistant at as early as 9 weeks for adolescents, regardless of the offspring diet, and have increased renal mitochondrial oxidative stress, inflammation, and fibrosis in the kidney, with overt albuminuria and renal structural pathological changes in adulthood or at 32 weeks. Recent data demonstrated that maternal high-fat diet induces also renal global methylation in the offspring at birth, and this was persistent till adulthood, supporting the role of epigenetics in fetal programming due to maternal obesity. Using reduced representation bisulfite sequencing, we identified genes that were differentially methylated in the kidney and blood of offspring of obese mothers at adulthood. Blue represents significantly hypermethylated genes, and red represents significantly hypomethylated genes. Overall, 21 differentially methylated genes were identified, 5 out of which were common to the kidney and the blood. Several of these genes have previously been shown to be relevant to the kidney. Since we demonstrated that high-fat diet and maternal obesity induce DNA methylation, we then wondered whether modulating DNA methylation can prevent or limit CKD progression due to obesity or due to maternal obesity. Hydrolyzing is an antihypertensive agent which has been in clinical use since the 1950s. It is currently used for the management of resistant hypertension and in hypertensive disorders of pregnancy. At low dose, which does not reduce blood pressure, hydrolyzing has a demethylating activity. So we aim to examine whether low-dose hydrolyzing can limit obesity and maternal obesity-related kidney disease. So first, to examine the effect of hydrolyzing on obesity-related chronic kidney disease. Animals were fed either high-fat diet or chow from 8 weeks of age. Animals on chow or high-fat diet were administered with vehicle or low-dose hydrolyzing in drinking water and culled at 32 weeks of age. Biometric and metabolic parameters were assessed in addition to markers of DNA methylation and renal pathology and function. Briefly, high-fat diet increased the animal body weight, kidney to body weight, and liver weight, in addition to retroperitoneal and epididymal fat weights, and these parameters did not change by hydrolyzing administration. High-fat diet induced albuminuria and glomerulosclerosis, both of which were significantly reduced by the administration of hydrolyzing. Fibronectin and collagen 4 were increased in the high-fat diet group, and both were significantly reduced in the hydrolyzing group. Obesity also increased the mRNA expression of the oxidative stress marker NADPH oxidase NOX2 and the protein level of 8-OHDG, a marker of DNA oxidative damage. Although NOX2 was not affected by hydrolyzing, the levels of 8-OHDG was suppressed by hydrolyzing treatment, suggesting that the protective effect of hydrolyzing may occur through reducing DNA oxidative damage. Interestingly, global DNA methylation was increased in obese mice versus lean mice, but this was significantly reduced even below control levels in the presence of hydrolyzing, confirming its demethylating effects. Using RRBS, we identified 109 genes that were differentially methylated in the kidneys due to high-fat diet, 47 of which were reversed with hydrolyzing. Pathway analyses revealed that hydrolyzing regulates genes involved in signaling pathways related to cellular metabolic process, kidney development, epithelial cell proliferation, and nephron development, and genes implicated in inflammation, cell death, and fibrosis. To examine hydrolyzing's effect on maternal obesity-related kidney disease, we administered hydrolyzing to normal or obese pregnant mice 3 times per week during gestation. Offspring were weaned on chow or high-fat diet, and animals were culled at 32 weeks of age. We had 8 groups, as you can see here. The first letter represents the mother's diet, and the second letter represents the offspring's diet. Example, HC is high-fat diet in the mothers and chow in the offspring. HH means high-fat diet in the mothers and high-fat diet in the offspring, or double obesity. The last 4 groups have gestational hydrolyzing. Postnatal high-fat diet induced significant adverse effects on biometric and metabolic parameters. Obesity and double obesity increased glucose intolerance, fasting blood glucose level, and induced hyperinsulinemia, insulin insensitivity, hypertriglyceridemia, and plasma-free fatty acid levels. Administration of hydrolyzing during gestation did not affect these markers, although data are not shown. Urinary ACR or albumin to creatine ratio was significantly increased in the offspring of obese mothers and in high-fat diet-fed offspring. And this was amplified if the high-fat diet-fed offspring were from obese mothers, so in a double obesity group. Gestational hydrolyzing administration protected the offspring from albuminuria. Serum creatinine was significantly increased due to double obesity, and this also was reduced in the presence of gestational hydrolyzing. Glomerulosclerosis was significantly increased in the offspring of obese mothers and in high-fat diet-fed offspring, and this was also amplified in the presence of double obesity. Hydrolyzing administration during gestation significantly reduced glomerulosclerosis in all the groups, regardless of the diet. Picosirus red staining showed increased collagen 1 and 3 deposition in the offspring of obese mothers and in obese offspring, regardless of the mother's diet. But hydrolyzing administration during gestation had no effect on Picosirus red staining. Tubular interstitial fibrosis was observed in the offspring of obese mothers, in high-fat diet-fed offspring, and in the double obesity group, as you can see here. 8-OH-DG, or the DNA oxidation marker, was also increased in the 3 offspring group compared to control, and this was reduced by gestational hydrolyzing, similarly to the tubular interstitial fibrosis, which was sort of not increased in the presence of hydrolyzing. Global DNA methylation also was increased in the 3 groups, and as you can see, this was also reduced in the presence of hydrolyzing in all the groups. So in summary, administration of hydrolyzing to obese animals improved albuminuria and glomerulosclerosis in association with epigenetic regulation. Gestational hydrolyzing limited the adverse renal outcomes of maternal obesity, offspring obesity, and the combination of both conditions. Our data support the repurposing of hydrolyzing as a renoprotective strategy in obesity and maternal obesity. So as a take-home message, we demonstrated that epigenetic regulation, specifically DNA methylation, has an important role in chronic kidney disease progression and may be used as a potential diagnostic marker for progressive kidney disease or progressive CKD. Early intervention at the onset of diabetes can limit chronic kidney disease progression. We also demonstrated that adverse in utero or postnatal environment can induce CKD in the offspring or pose a risk for future disease development, and intervention during the prenatal or postnatal period can prevent or limit chronic kidney disease development. Finally, I would like to acknowledge everyone's contribution who have assisted me in the completion of this work and my funding bodies. Thank you for listening.